The previous two speakers have done a wonderful job of explaining why what I'm going to tell you is so difficult to accomplish. But I'm also going to focus on screening rather than diagnosis. And as I'm sure you know far better than I, traditionally screening has been done with a chest radiograph. But there's been a relatively slow transition to using CT, low-dose CT in particular, for screening because it seems to do a better job. And by seems to do a better job, I'm basing that on this study that I participated in. My lab was one of the data coordinating and quality assurance centers for the National Lung Screening Trial. And the result of that trial, after about a decade, was, as you can see from these two key graphs, that the number of cancers detected using low-dose CT was greater than in the radiograph arm, and there was a decrease in mortality due to earlier detection. And that decrease in mortality was about 20%, which is the primary reason why we began this transition to using CT. This study, and it's already been mentioned by the first speaker, had another interesting property, and that is that the false positive rate, I'll get my tool in a minute. The false positive rate was between about 95%. That makes this a little bit more challenging, but of course there's really little penalty for a false positive in that we're just going to go on to a diagnostic workup where a false negative could be fatal. So the false negative rate in this study was quite low, at about 6%. But the question that we are trying to address is, can we do better than this using quantitative image analysis and machine learning techniques? And in particular, the name that's given to that approach these days is Radiomics, thanks to this foundation paper by Hugo Ertz and Bob Gillies and their colleagues, where they coined the term radiomics and laid out this basic pattern of extracting information from images, first by segmentation and then identifying features, characterizing those features, and representing it in a new way, representing the imaging data in a new way. Now, as it turns out, these ideas are not new. And I hear a lot of discussion about, well, AI and machine learning are going to make radiologists obsolete. Well, back in the 1960s, Willem Lodwick published a paper where he introduced the concept of computer-aided detection, computer-aided diagnosis, CAD and CADX. The tools that we use today were developed largely by people in the CAD world in the 1990s. This is a paper by my colleagues at Georgetown, Sung Ki Moon's lab, and Ben Lowe was one of the developers of some of the deep learning algorithms that we still use. Kunio Doi's lab and others were very active in creating these tools, and they resulted in commercial tools that probably many of you have used. So you've already been using these techniques if you've ever used a CAD algorithm. What's important, I believe, is this part of the picture. What we are doing is representing images in a new way, in a feature space, and by doing that, we have the ability to combine those features with features derived from genomics and proteomics and other types of analyses so that we can begin to look for mechanisms across scale by finding patterns in these joint feature spaces. So I'm going to come back to that topic briefly a little bit later, but I think that's what makes what we're doing now a bit different. This is a research tool predominantly. Now, we can also use it to build a new type or a new approach to aids for the radiologist. And that's really what most of my talk is going to be about, the development of assays that remove some of the burden of the radiologist, and I'll explain what I mean by that. So first, what is this thing that I just described called a feature space? So following Bishop, a feature is a measurable property of a phenomenon or an object that's being observed. So it's a measurable property of something in an image. And in medical imaging, there's been a bit of confusion because sometimes the region of interest itself, a lung nodule, is considered to be a feature, but it's really the measurements of the properties of that region of interest that we deal with and what we call features. So properties such as the volume of the object, its shape, its texture, how uniform or non-uniform the grayscale is in that region. These are features, and we extract those features using mathematical or quantitative imaging techniques. And the entire set of features of interest define a feature space. And this tends to be a fairly high-dimensional space. We use about a 50-dimensional feature space in one of the examples I'm going to show you. And it's in that high-dimensional space that we have the ability to use more advanced machine learning tools and to combine it with other types of information. So there are a couple of different classes of features, and I'm going to focus on this one because it's the one that's most widely used. We call them engineered features. So this is a lung CT. We've identified an object of interest. We segment that object, we separate it from the background, and then we calculate a number of features that describe that object. And these features, we didn't invent them. Radiologists have been using them for years. What we do is define a quantitative metric that characterizes the thing that you've been using all along. So size, this is just showing something like a resist measurement. Actually, we measure size in more esoteric ways. Shape, how different is this from a sphere or other regular property, polygon. Boundary condition, this is very important. It's how diffuse is the object of interest. Tends to be roughly correlated with how aggressive the cancer is. Texture, which I've already mentioned. And what we do, and then various other intensity or distributional type properties. And the purpose of this is to generate this matrix of numbers, which is the feature space representation of all such objects in this image. So each column is a vector, or a feature vector, and this defines the feature space for this set of data. So these engineered features are important because I can name them, right? I can tell you what characteristic I'm looking at. In fact, we're developing ontologies of features where we use the mathematical expression of how I calculate it. So, for example, my friend Sandy Napel uses a Gabor filter bank to measure texture. We use wavelet coefficients, but we can precisely define what we mean by texture. And that's important, and that's why these features are useful. So, the problem, of course, is that as with many areas of science, there are a lot of different ways of measuring these things. And so Jayshree Kapathy-Kramer and some other colleagues from the Quantitative Imaging Network program at NCI that I'm also involved in, did a survey of the literature and came up with 830 lung nodule features, which they've classified, and the classification happens to be a kind of the classes that I've just been showing you. So there's a lot of different ways of measuring this, which makes it a little bit more difficult to understand, but we use our own feature space representation, our own set of features that were derived from this experiment done in my lab at WashU by David Gerada and David Polite, where they actually looked at different resolutions of the same object. So we collected the raw sinogram data from CT scans, and then reconstructed at small fields of view, high resolution, then made 3D volumes of objects of interest. And so we showed radiologists the same images, but at different scale and in 3D versus 2D. And there was a wide variance in what was considered to be malignant or benign, as you might expect, because of the changes in resolution and representation. That was an interesting experiment, but not what I wanted to talk about. What came from that was a set of 121 features that we use. I'm showing you the three-dimensional ones, which turned out to be more useful, but they're also a two-dimensional set in that collection. And it turns out to be pretty much a superset of what most other people are using. So this is our feature space representation. So the first experiment that I want to just talk about was conducted with my colleague Shujin Wang, and at the time our student Jason Causey, now Dr. Causey. The question was, can we make a machine learning algorithm that can classify lung nodules as well as a radiologist? And we started with a different data set, which was the Lung Image Data Consortium's data set, because it's a wonderful set for training an algorithm. It's over 1,000 cases where a panel of radiologists classified, first of all, segmented all of the objects of interest and then classified them on a scale of 1 to 5, 1 being high probability of benign, 5 being high probability of malignant, and then the various other probabilities in between. And so this gives us a wonderful data set to train our algorithm. And so what we did was take that picture that I just described of extracting, segmenting out these objects, extracting the features, and we ended up with about 50. We used the 121-dimensional feature set, as I said, and then a feature space reduction, principal components analysis, to arrive at 50, which explained most of the data, and then a random forest classifier, a simple machine learning algorithm that did a pretty decent job of classifying the data. Now, because this, for these kind of algorithms, was a relatively small data set, we tried a number of different variations on this, and the different types of training, different approaches to show that we didn't overfit the data. And in general, we got a pretty reasonable classification accuracy. It's a fairly simple problem, but it was useful for us in order to train algorithms and to begin to explore this. Now, I mentioned the idea of combining information across scale, and so I want to just give you a teaser of some of the work that's using this particular pipeline that we developed, and some work that's been done by my colleagues at Stony Brook, Joel Saltz and his team, where they were developing the pathology equivalent of what we're doing, and developed tumor-infiltrating lymphocyte maps, or TIL maps. And these are maps, if you're interested in, they're available from the Cancer Imaging Archive. You can search for them, or that's the link. And so what we're going to do is, their original work was using the TCGA data set. We've got some new data from the CPTAC program, a couple different cancer types. And so we're generating the radiology features and combining them with the pathology features to begin to look at what we can do with this joint feature space. And this is something that we hope to demo at RSNA this year. But that's just a little teaser to say that's where I think the main value of radiomics is in this context. But we moved on from this engineered feature space to the next level, which is using deep learning, again, algorithms that have been around for a while, where we have the ability to learn the features as well as doing the classification. So in the engineered features, we need a lot of help from a radiologist. In this approach, we simply segment lungs, and we have a classification from outcomes. This person developed cancer, yes or no, and eventually what type of cancer. And so we train a deep learning algorithm to be able to extract features of interest and then classify based on those features. And so how does that work? I mean, maybe you're all familiar with this, but it all starts with a artificial neural network. And the idea of such a network is I put in an input pattern, and there are one or more hidden layers. In the case of a deep learning algorithm, there are a lot of hidden layers. And it presents an output. And you can think of the output as definitely benign, definitely malignant, and we don't know, which is the way we're looking at it. And so what happens is if you have a training set where a human is labeled, given us the right answer, then you feed in the input, and at the output, you get an answer, you compare it to the right answer, and you generate an error. And there are clever algorithms. The one that most people use is called backpropagation, where you distribute that error to these W's, weights. And so the way the network works is you take the input and multiply it, or the output of this node, multiply it by the weights. That becomes the input here. There's some function in the node that sums them all up and has a rule for deciding whether or not it fires. And that's the way this works. So learning is really changing the weights. So now we go to a more complicated model of the same kind of thing. This is a convolutional neural network, which I'm sure you've all heard about, where what we're doing is starting with an image, and we're applying a digital filter to that image. Each digital filter stage extracts features. And the power of the convolutional neural network is that this stage extracted features, this stage extracts features from its output, etc., rattling down the line. And at the end, we have a fairly normal neural network like the one I just described. The backpropagation, however, changes the weights, and the weights are the filters. So this learns to extract new features. And that's where it gets its power, is when you train it, it's learning what it feels is important in these images in order to classify them correctly. And so we used that algorithm, that approach, on the same data set and came up with a reasonably good result. And Jason said, well, what if we combine the two? And I said, well, they're both giving good results. I don't see why we would get anything different. And it turns out his question was what made this an interesting paper, because in fact, we did get a different answer. We got a better answer. Each approach had learned something that the other hadn't. And the combination of the two provided more information, a better classification than either of them separately, which we found quite interesting. So that was an interesting approach to get us to a certain point in our development, which was really to begin to develop this. And about that time, there was a challenge competition. It was called the Data Science Bowl Challenge. We were not allowed to compete in the challenge because we provided the data, but we were allowed to participate. And we developed an algorithm that was based upon this, but with a lot of other stages built and piled in front of it, that actually did as well as the algorithm that won. But the problem that we were trying to address is the real one. Not this toy problem I've just been describing, but the real one, which is, can I take lungs and output a risk score of whether or not this person is likely to have cancer? So can I automate the screening process? Not necessarily to do the screening all by ourselves, but rather we wanted to focus on, can I really find the normals? To take those off the table. The ones that we're really certain are normal, leaving a much smaller load for the radiologist. And the ROC curve demonstrates that we did reasonably well. In fact, as I said, we did better than as well as the other algorithms. Still not perfect. Work is still not done. But one of the things that we'd focused very heavily on was this problem of a high false positive rate. And in this case, our false positive rate I should have mentioned, the Data Science Bowl Challenge used a number of screening data sets, some of which were from the National Lung Screening Trial and supplemented from other sources. So we took a completely independent set of data from the National Lung Screening Trial. Unfortunately, we're the only ones that could do that because we built both data sets, so we knew which ones were independent. But we took a completely independent data set, and that's what we tested this on to see how well it generalized. And so these are the results of the generalization experiment, not the original Data Science Bowl Challenge. And so comparing to the NLST's 95% false positive rate, we got a false positive rate of 2%. We were very happy about that until we calculated the false negative rate, which wasn't so good. And that means that we haven't tweaked the algorithm quite correctly yet. We focused too much on solving problem A and, of course, ended up with problem B. But I think we demonstrated that the approach has some merit, and we are going to continue to develop this. We unfortunately hit the problem of we've run out of data sets on which to test it that are independent, so we're now trying to accumulate those as we tweak the algorithm. So what I want to impress upon you is first of all a couple of things, that this idea of AI, machine learning, taking over radiology, don't worry about it. You've already been using this stuff for a long time. You already know how to use it, and these are simply a different approach to allow us to perhaps find new mechanisms to better understand features and knowledge that we already have, or develop some new knowledge from those experiments, and perhaps to create tools that I call assays that would be interpreted by a radiologist to say, yeah, we're reliable in taking away the easy cases, the normals, leaving the more difficult and challenging things that take into account all the factors that we heard from the first two speakers. That's left to the human, because humans have intuition and algorithms don't. And in fact, there's a wonderful proof, mathematical proof, that algorithms can never have intuition. So I will leave you with that thought, and acknowledge both the team relevant for this research and our funders. And thank you.